Okay. All right. Great. I can see that the stream is on. So whoever is already online in this session. Oh, I have some echo. Okay, I hope now the sound is good. My name is Petar Lambrev, and I am here at my office in the Biological Research Center in Szeged. And I'll be talking about ultrafast two-dimensional electronic spectroscopy. Now, this is a technique, ultrafast technique, obviously, that was invented maybe sometime around 20 years ago, but since then has very rapidly gained popularity in the research communities in photochemistry, photobiology, and especially in a photosynthesis, which is the area of our group research. So I will be giving references to photosynthesis throughout this presentation. Now, here is the outline. We will start, I'll try in this time to introduce mostly the technicalities and principles of the method, but we will start easy with the basics of electronic spectroscopy, which is, of course, very familiar to everybody. And then we will add on the time dimension and talk about transient absorption spectroscopy. Then add another frequency dimension to make a two-dimensional spectroscopy. Then we will see how this can be done in practice. And if time allows, maybe one or two examples, I would show you of measuring energy transfer in photosynthesis with this technique. So let's start with the electronic spectroscopy. And what is it? Or actually, we can ask, what is spectroscopy in the first place? Now, spectroscopy, of course, is a way to understand the properties and behavior of molecules by watching, by observing their interaction with electromagnetic radiation, be it light or microwaves or X-ray. So what kind of interaction are we talking about? Well, the basic, we have absorption, we have emission, we have scattering or reflection. Now, we will mostly talk about absorption. And absorption, what is absorption? So we know light is electromagnetic wave, which we can also be thought of as a ray of photons. And when a photon hits a molecule or an atom, it could be absorbed. What does it mean? It means that the transfer of energy happens. The photon energy is transferred to the atom or molecule, and it goes into a higher energy state. So the opposite can also happen in emission, where the molecule or atom will go relax back from the higher to a lower energy state, and then the released energy will be emitted as a photon. So depending on the energy of the photon, the absorption can cause different changes in the molecule, right? If we start with the lowest energy photons of microwaves or infrared radiation, they cause just a molecular rotation or vibration. But we are talking about electronic transitions here in electronic spectroscopy. That means when higher energy photons of visible or ultraviolet light interact with the molecule, the electrons can jump from one orbital to a higher energy orbital, or we say that the molecule enters a higher electronic state. Of course, if the energy is even higher, like in ionizing radiation, X-rays, the electron can be kicked out altogether from the molecule, then we have photoionization. So we're focusing on basically UV visible spectroscopy. Now, the simplest kind of electronic spectroscopy that everybody knows is spectrophotometry. We shine light of certain color on a sample that is maybe solution of some molecule, and we are measuring the intensity of the transmitted light relative to the intensity of the incident light. That is what we call transmittance. Now, the negative logarithm of that quantity is the absorbance, and we know from Bayer's law, the absorbance is proportional to the concentration of the absorbing molecule. So we can detect the molecule, we can determine its concentration, we can use it even for mixtures of different compounds. Now, very useful technique. If we measure the absorbance as a function of frequency or wavelength or color, then we get an absorption spectrum. So how do we do it normally? We have a monochromator that can tune the color of the incident light, and we gradually change the color, and we measure step by step the absorption, and we construct the absorption spectrum. But there is another way. If we look closely in the monochromator principle is this, that we shine the broadband light on a dispersive element, a grating or a prism, that disperses the light onto a slit. But if we remove the slit or aperture here, and instead put an array detector like a camera, a digital camera, then we can measure all the wavelengths at once. And then we have not a monochromator, but a spectrograph. And this is very useful because we can, in this way, measure the entire spectrum at once. And also, one can make very small miniature spectrometers that are now very widely available. You can put them on a drone and do great things with them. So let's go back to the physics a little bit of the process. We said the molecule which absorbs the photon enters a higher energy state. But what are these states? Of course, these are eigenstates or solutions of the time-independent Schrödinger equation. So every eigenstate has an energy associated with it and an eigenfunction which defines the wave function of the molecule or the system that we are looking at. So the absorption spectrum is just a neat reflection of the electronic structure of our molecule. It has bands which correspond to electronic states, and the band position in frequency corresponds to the energy of the states. The band intensity corresponds to the probability that the transition will occur, which is something to do with the dipole strength or the square of the transition dipole moment of that particular state. We also have the width of the transition, which tells us about the spread of the energies or the width of the energy band, which is controlled by the things that are called homogeneous and inhomogeneous broadening effects, which maybe we will talk about later. Now, let's see what happens in the molecule when it is in the excited state. Of course, most of the time, any molecule that is excited will go back to the ground state via one or another pathway of relaxation, or it can initiate a photochemical reaction, which is the basis of photosynthesis or vision or entire photochemistry. One thing quite interesting is that it can do excitation energy transfer. That means if there are two similar molecules nearby so that they can feel each other, the excited acceptor molecule A here can relax to the ground state. At the same time, the other molecule B, which is a donor molecule, will be excited, and then the excitation effectively jumps from one to another. This is a familiar resonance energy transfer mechanism or Förster energy transfer mechanism, which has very many uses in nature, but also in scientific research. Now, another thing that I'd like to remind, which is very important, is quantum coherence. So can the molecule be at the same time in the ground state and in the excited state? Well, think for a second on that. Maybe someone can write me yes or no in the chat. All right, I will write yes. Of course, these eigenstates are quantum states, and we know that from quantum mechanics that any solution to the Schrödinger equation can, any two solutions can be combined, and any linear combination is also a solution. So every combination of eigenstates is also a valid quantum state, and it is called a quantum superposition state or a coherent state. This is a fundamental property of quantum systems, and it's very important for their behavior. So the coherent state is a superposition, a linear combination of the ground and the excited state in this case. And unlike the eigenstates, it's not a stationary state, it's a dynamic. It has this here term, if you can see this complex exponential. That means that this state, the wave function, will oscillate, and it will oscillate with a frequency, omega, which corresponds to the energy difference between the two states. So this is something to remember because it's crucial for any kind of coherent spectroscopy. Now, we are talking about dynamic processes here, energy transfer, coherence, and so on. Now, if we want to measure any kind of such dynamic processes, we go into the domain of time-resolved spectroscopy, and where we make movies or snapshots of the molecule in different periods of time. So what periods of time? It's worth to look at the ruler of timescales of biological processes. It spans a very large range of timescales. You start from enzyme reactions that can go to seconds, but if you go shorter in time, you can have photosynthetic electron transfer, for example, or nerve impulses or ion channel gating that go from microseconds to nanoseconds. Now, it pays to remember that a nanosecond is 1/1000th of a second, very short, obviously, but a nanosecond is kind of eternity compared to the timescales of photosynthetic charge separation or exciton migration, which happens in between femtoseconds and picoseconds. Now, femtoseconds are very short periods of time. So in ultrafast spectroscopy, usually we are interested in processes that occur on timescales from femtoseconds through picoseconds to nanoseconds. All right. So how do we do that? Well, the one very powerful and very widely used technique is transient absorption, time-resolved transient absorption spectroscopy, where we create a transient non-equilibrium state in the system that can be an excited state and can be created with a laser pulse. And then we will probe the system in that excited or transient state. So this is essential method, as I said, in photochemistry, photobiology, and it can reveal information about the excited state energies, probabilities of transition, the decay and the lifetime of the excited states, the energy transfer, photochemical reactions, and so on. All right, so how does it work? Very briefly, well, first we need a laser that can provide ultra-short pulses, and then we focus two pulses on the same spot of the sample. The first pulse that we call pump is the one that creates the excited states. We are not interested in measuring the absorption of this pulse. It's just to condition our system. Then after a time, we send another second pulse, which we call the probe, on the same spot, and we measure the changes in absorption of the system that were induced by the pump. So actually we need to make two measurements and take the differential absorption of the probe pulse with pump and without pump to subtract the background absorption. Okay, in a nutshell, and if we have a spectral detector, we can measure spectra, transient absorption spectra. So what do we see in the transient absorption spectra is usually three main kinds of signals. One main signal is called ground state bleaching. That is because the first pulse will excite the system, and then there are less molecules in the ground state that can absorb the light. So we have a negative absorption or bleaching, transient bleaching. Another negative signal is the stimulated emission. That is, the molecule is in the excited state, and then we send in the probe pulse. The probe pulse can force the molecule back to the ground state, and the excess energy is emitted. So that is stimulated emission. Or the molecule that is excited can absorb the probe pulse, photon of the probe pulse, and then the molecule goes to another higher energy excited state. So this will be excited state absorption or photo-induced absorption. And then these three signals will combine to make our transient absorption spectrum, which is a signature of the molecule in the excited state. Now, technically, how this is done? Well, until some years ago, this was a very complicated technique that was available in a few labs only, but nowadays it's really very common, and you can buy a ready-made femtosecond instrument from a number of manufacturers, or you can buy the off-the-shelf components to build one yourself. The main component, and obviously, is the source of the ultra-short pulses. So you need a mode-locked laser. That's also the most expensive part of the system. Now, if you have a mode-locked laser, you can take the beam and split it into two paths, the pump and the probe. So basically what you need, usually the pump beam is a narrow band tuned, maybe with some filtering or nonlinear processes to a wavelength that is tuned to your molecule of interest. And the other thing you need to do is to modulate the pump with an optical chopper or something. That means to switch it on and off, because you have to take two measurements and take the differential absorption with and without the pump. And all you need to do then is to focus it on the sample. The probe then is passed through a translation stage, or what's called optical delay line, where we can vary the delay of the probe pulse relative to the pump. The probe is usually also made into white light, so that it is broadband, so that we can measure the entire absorption spectrum. This is often done just by focusing it on some kind of crystal, for example, sapphire, and then it becomes white light. Then you focus it on the same spot on the sample, and then we have to pick the probe, the transmitted probe beam, and focus it onto our spectral sensitive detector. So when we do the measurement, we get data as a function of wavelength and as a function of probe time or delay time. So this allows us to measure, to analyze the time result spectra at different time points, or we can take in the other dimension the time dependence of the absorbance at different wavelengths. And this provides us already a lot of information of what is happening in the system over times that can be from here 100 femtoseconds to 100 picoseconds. Now, this is already quite powerful, but we can make it even more powerful by adding one more dimension. And so now we are ready to talk about two-dimensional spectroscopy. Now, the crucial aspect of two-dimensional spectroscopy is in its name. Instead of one dimension, one frequency dimension, it has two frequency dimensions. So what are those? Now, in transient absorption, we frequency resolved the absorption of the probe beam. In two-dimensional spectroscopy, we have frequency resolved response to the pump and the probe together. And so we have a pump or excitation frequency, and we have probe or detection frequency. So in other words, a two-dimensional spectrum is a frequency-frequency correlation map that correlates our pump and our probe frequency. And like in other two-dimensional spectroscopies, like in NMR, for example, if we have cross peaks, then we know that two frequencies are coupled, or we know that two energy states of our molecular system are interacting or coupled with each other. Okay, so this is a extremely powerful technique. In fact, you could say that the information that you can get from a two-dimensional electronic spectroscopy contains any kind of information that you can extract from any other kind of third-order spectroscopy. What is a third-order spectroscopy? We will see in a moment. So how do we do the two-dimensional spectroscopy? How can we measure two-dimensional spectra? The simplest way is you can think of the two-dimensional spectrum as just we repeat a number of transient absorption measurements with different pump colors, with different pump frequencies. And then we can stack them together vertically to make the two-dimensional spectrum. This is possible and it's doable and people do it. So you can take a series of measurements varying the color of the pump and then stack them together. The problem is that it's laborious, time-consuming, but even bigger problem is that you hit what is called the transform limit. So the transform limit tells you that you cannot have infinitely short pulses that are also infinitely narrow in frequency. If you have very short pulse, it has to be broad in frequency. So you have to compromise a trade-off between high time resolution and high frequency resolution. So how do you avoid that? The solution is broadband Fourier transform two-dimensional spectroscopy, or this is the real two-dimensional spectroscopy, what people usually mean. And again, the essence is in the name. Instead of narrowband pump pulses, you use broadband pump pulses, and you measure the signal in time domain, and then you do Fourier transform to get to the frequency domain. So what does it mean? It means we send not one, but a pair of two pump pulses, and now they are broadband, so they can be as short as we like. They can be a femtosecond short. So then we will have a time period, which is called coherence time, between the two pump pulses, and we scan over this time period, and then we will Fourier transform the signal as a function of coherence time to get frequency. So in one type of signal response, which is called the photon echo, the first pump pulse will create, if you remember, it will create coherent state. That means the system is halfway in the excited state. It is oscillating back and forth between the ground and the excited state. And this coherence lives throughout the coherence time. Now the second pulse, if it arrives in phase with the oscillations, it can also interact with the sample, and then it will drive the full excited state, convert it to a population. So from then on, we have the sequence that is common to pump probe spectroscopy. We have a waiting time or delay time, and then we send the probe, and we see how the system, what's the state of the system after this delay time. So the probe will also create, in principle, a coherent state that we detect, but this is, you don't have to worry about that. We just measure the signal at the time of the probe. So in reality, we get the signal as a function of two frequencies, the pump frequency and the probe frequency, and one time delay. So this is the two-dimensional spectrum. Now, what is it good for? Well, it's good for a variety of things. That means you can extract different kinds of information from a two-dimensional spectrum. We are very interested in resolving energy transfer, and two-dimensional spectroscopy is extremely good in that. Energy transfer means you have the two molecules which have slightly different frequencies. So you pump at the frequency of one molecule, it will transfer the energy to the other. You can probe then at the frequency of the acceptor molecule. What you will see is a cross peak on the two-dimensional spectrum that connects the frequency of the donor and the frequency of the acceptor, so you know what's going on. And also this is dynamic, because this is, of course, a time result spectroscopy, so you can follow the intensities of the cross peaks in time, and then they will tell you the kinetics or the rates of energy transfer. Okay. Now, energy transfer can be reversible, and usually is reversible. That means it can go forward, it can also go backward. The ratio of the forward and backward rates is defined by the detailed balance or the Boltzmann distribution. And in two-dimensional spectroscopy, we can separate these two very easily because we have separation of the pump and the probe axis. So we have a cross peak that corresponds to the downhill energy transfer. We have a symmetric mirror image cross peak that corresponds to the uphill transfer. It's very useful. Another kind of information, and very important, is the line shape analysis. So as I mentioned that the lines, absorption lines, are broadened by two processes, generally termed homogeneous and inhomogeneous broadening. In regular spectroscopy, they just add together. In two-dimensional spectroscopy, you can separate them. Homogeneous broadening is when all the molecules fluctuate around their mean energy. So this creates kind of randomness, and it broadens the peak shapes in both directions. But inhomogeneous broadening is when you have an ensemble of molecules, each with a slightly different energy because of inhomogeneous environment. And then that creates such elongated line shapes because there is correlation or memory in the frequency. That means each molecule knows its own frequency. So when you excite that, you also probe it. And there is the intermediate situation where a molecule knows its frequency initially, but over time it will fluctuate and it may forget it. So we can also measure that. That is called spectral diffusion. And another type of information that you can follow in two-dimensional spectroscopy, and this is actually the main reason why the technique became so popular, is that you can follow coherence dynamics. That means the pump pulse can create coherent superposition of several eigenstates. And when that happens, this coherence can survive over the waiting time, and this can be observed by periodic oscillations of the cross peaks in two coupled states, let's say. So you can measure those oscillations, you can get the frequency of the oscillations, that will tell you which states are coupled. And this has been observed in a number of systems, especially light harvesting complexes of various photosynthetic organisms. and created the idea that this quantum coherence is an intrinsic part of photosynthesis and also other actually biological processes. Okay, so I hope that you are now familiar with the basics of the method. I would like to just run a little quicker through the technical implementations of two-dimensional spectroscopy. Now, there are two main approaches which are used nowadays. The most common is the so-called non-collinear or boxcar geometry. This is where we have the two pump pulses and the probe pulse, and also a fourth called local oscillator pulse, all focused on the sample at different incident angles. And that's then the signal that we are looking for, the photon echo signal, travels in a so-called phase matching direction, which is different from the incoming pulses. So we can measure it free from the background of the pump or probe pulses. This is the most versatile of the two-dimensional spectroscopy methods. It gives full control over what we are measuring, but it's the most demanding, and it's very easy to introduce artifacts and distortions if it's not done properly. The other common method is a partially collinear method, where we have the two pump pulses going collinearly, just one after another, and then the probe comes at an angle. So then the photon echo signal will travel in the direction of the probe. And this is exactly how we measure transient absorption. And in fact, you can upgrade your transient absorption spectrometer very easily to a two-dimensional spectrometer just by converting your pump pulse to a train of, or a pair of two pulses. But you need a control delay and phase between them. So there are two fundamental differences of how to get the signal separate from the background. In the non-collinear approach, this is natural. As I said, the signal travels in a phase matching direction that is different from the incoming pulses. But you need to make two measurements to get the full spectrum. You need there are two phase matching directions, the so-called rephasing and non-rephasing. You have to combine the two very carefully to get the spectrum. In the pump probe geometry, you also do two measurements, at least. It's called phase cycling. You have to flip the phase of the pump pulses, and that will flip the phase of the coherence. And in this way, you can subtract all other signals which do not have a phase dependence, so the background signals. This is an example of the non-collinear setup that is developed by Graham Fleming and nowadays used in many labs around the world. I won't go into detail, but basically you have two pulses, you separate them into four, and then you introduce delay stages so that you can control the delay between each. Then you focus them on the sample, you have a screen, and you only let the photon echo signal to the spectrometer. Now for the pump probe geometry, as I said, you can upgrade your transient absorption spectrometer, but what you need is a device that makes a pair of pulses, and that is a pulse shaper. And there are several now manufacturers that give ready pulse shapers useful for two-dimensional spectroscopy. They're not cheap, but they work really well. This is one example of two-dimensional spectroscopy setup. Won't go into the details, but it's really just a transient absorption spectrometer with one addition, this acousto-optic pulse shaper, which makes from one pulse two pulses, and then you can control the delay. Okay, there are various other ways but we will skip the rest of technicalities, because I would like to just use some time to show you one or two examples in photosynthesis. Okay, so it's no a secret and no need to repeat how important photosynthesis is, because it basically drives all the life. All the life on Earth runs on solar energy, which is converted to chemical by the phototrophs, and like plants. And that happens in the chloroplast thylakoid membranes, where you have the two photosystems, the photosystem 1 and 2, which capture the light and drive electron transfer and proton transfer to make ATP and electrons for carbon fixation. So we're interested in these photosystems. They are packed full of chlorophylls, and chlorophyll is a derivative of porphyrin. It's the photochemically active molecule in photosynthesis. Now in the photosystems, like this is the photosystem 2 super complex, this is the protein. As I said, it's packed with chlorophylls. Most of these chlorophylls are so-called, these are all chlorophylls and carotenoids. Most of them are antenna chlorophylls. That means they simply transfer the absorbed energy to the middle here where the photochemistry happens. And the entire system depends on the efficiency of this transfer. So the two-dimensional spectroscopy, as I said, is very useful for measuring energy transfer. In fact, the first demonstration of two-dimensional spectroscopy was in a photosynthetic light harvesting complex called the FMO complex, which is a fairly simple complex. I won't go into the detail, but it has only 7 pigments, so they have 7 excited states that give 7 peaks in the two-dimensional spectra. Now, another very common, actually the most abundant light harvesting complex is the plant light harvesting complex 2. It has 14 chlorophylls. These are chlorophylls A and B, and because it's so important, it has been studied for decades, the energy transfer. But the most detailed information came from two-dimensional spectroscopy, and I just want to show you here. These are 2 spectra of light harvesting complex 2 measured at very low temperature of 77 Kelvin. You can see here at very early delay time, most of what you see is diagonal peaks, which correspond to the different excited state energy levels in the system. And once you give some waiting time, let's say 100 picosecond, all the higher energy excited states have disappeared, and the energy has transferred to the lowest energy, long wavelength chlorophylls, which are the terminal acceptors of energy. But of course, this is time-resolved technique, so we make a movie and we can follow how the energy disappears from the short wavelength forms and the cross peaks of the long wave forms appear. So then we can analyze those data in terms of various models. The simplest is what's called global analysis. We fit in multi-exponential function to the kinetics, and then we get lifetimes of energy transfer, characteristic lifetimes, and then what is called decay-associated spectra that show us different states that disappear and different states that appear on certain timescale. So you can get a rough picture of what is going on in different timescales. But that's relatively crude. You can do a much more sophisticated modeling where you have variety of excited states, and you fit all the traces in the 2D spectra. So you can reconstruct the original two-dimensional spectra, and with this you can get the energies of the excited states, their widths, homogeneous and inhomogeneous, and of course the rate constants of energy transfer, basically the entire kinetics of the system. Okay, so I will just repeat the main points of this technique to remember about. The two-dimensional spectroscopy reveals information about, well, couplings between your chromophores, the population dynamics or energy transfer between your molecules, coherence dynamics, that is exciton, vibrational, there are various kinds of quantum coherence. You can separate homogeneous and inhomogeneous broadening and measure them in dynamics, the dynamics of spectral diffusion, and it allows you to separate the bidirectional uphill and downhill or forward and backward energy transfer and thermal equilibration in complex systems. So just at last, I want to advertise that we, in collaboration with the ELI, are building a two-dimensional spectroscopy station that we think very soon will be able to welcome users to the ELI with their own samples and scientific projects. So thank you very much. This is all I had to say. And I can see now that there are a few